Can somebody confirm you can hear me?
Yeah, we
Yes,
agree.
yes, we can.
Great. All right, before we begin, are there any questions? Not hearing anything, so let's begin. Sharing my screen now. Oh, let's optimize. So today is October 10th, and we are supposed to be ending chapter 2 and beginning chapter 3 today. I don't think we ended chapter 2. Let me see that. Oh, yes, we did. We talked about hydrogen bonds. All right, so we're going to start chapter 3 today. Did anyone have any trouble with the quiz, meaning getting it to run? There will be a lab later on today in the lab, lab 4. All right, as is normal, I show you an introductory slide for chapter 3, observing microorganisms. The major goals and a rough outline are shown here. Know the terms of this chapter. What is resolution, refraction? What are stains? What are basic and acidic dyes? What is a smear preparation? What is a mordant? And what is peptidoglycan? Any questions about any of these terms? Now remember, when you're going through, these are only the major goals. If you want to know all of the objectives, you need to take a look at, let me see if I can find it, the study guide for chapter 3, and it'll have all of the objectives. One reason for doing that is all of the quiz questions are covered in the objectives, meaning all of my quiz questions have an objective. Now, where did that go? There it is. So there are 11 objectives for this chapter. Now, some of them, of course, have more than one question in it, but let's go ahead and close that because I'll get noise when an email comes in. And while I'm here, I can open up chapter 2. All right, chapter 2. So, number 2, understand the sizes of organisms and the properties of microscopes. Understand the major differences between the light microscope and the electron microscopes. 3, understand why staining improves resolution. And 3A, know the 3 types of stains. There are simple stains, differential stains, and special stains. And then 3B, understand the gram stain in depth, its procedure, and the basis of its differential staining. Any questions about these goals and rough outline? We're going to be talking about units of measurement and the metric system. You need to know the metric system because science and medicine both use the metric system. Let me try to blow this up so you can see it better. You do need to know most of these metric units, like you need to know the kilometer. It is 1,000 meters. You need to know the meter. It is the basic length in metrics for the length. You need to know, you don't need to know a decimeter. You do need to know a centimeter. That's 1/100th of a meter. And if you have a ruler on it that has metrics, the centimeter is on it. The millimeter, you need to know that that's 1/1000th of a meter, and that's the smallest unit on a ruler. You should know a micrometer. That is 1/100, no, 1/1,000,000th of a meter, or 1 to the 10 to the -6 meters. And this is the size of microorganisms. So when we see things under the microscope, we'll be talking about micrometers, how long they are in micrometers. And most bacteria are between 1 and 2 micrometers in diameter. A nanometer is the size of things you see under the electron microscope. So you should know a nanometer, and that's 1/100000th of a meter, or 1 times 10 to the -9th meters. A picometer you don't really need to know. Any question about any of that? Now in metrics, you just switch the base unit. There's a meter for length, a gram for weight or volume, and a liter for, excuse me, it's not volume, it's weight, or what the heck is that, space? That's not right. Mass, it's mass or weight for gram. And then for volume, it is liter. And then you simply use the prefix. So there's a kiloliter, a deciliter, a centiliter, with the basic unit. Any question about any of that? Now, the metrics are easy once you get to know them because everything is based on a scale of 10. Um, you don't need to know a decimeter, but that's 1/10th of a meter. A centimeter, you should know, is 1/100th of a meter. And then switching between different metric units just means switching your some factor of 10, like moving from a meter to a kilometer. You just uh, divide the meters by 1,000 to get 1 kilometer. Any questions about that? You are expected to know the metric system for the quizzes. You can see the study guide. I've got a metric quiz online to help you practice. You do not need to know the metric system and then converting to the English and vice versa. That is much harder. Any question about any of that? All right. So you do need to know the metric system because science and medicine use it. And here's the other reason why you should know the metric system. This map is showing you all the countries in the world that do not use the metric system. And two of these countries are actually switching over to using the metric system. The United States is actually one of the countries that is switching, but we are going at a snail's pace in switching over. I think the switch passed Congress when I think it was Andrew Jackson was president. So I think it was the third U.S. president. And if you notice the signs, like the road signs telling you how fast to go, they'll have in miles per hour in big letters, and then underneath it, at least this is in Oregon. I don't know if that happens in Washington, but it tells you I used to live in Oregon. Uh, Underneath it, it'll be metric in smaller print. And then on the speedometer, it'll have metric on top and not metric, English on top, and then metric on below, at least on the new cars now. Any question about any of that? All right, so you should know a little bit about the typical size of microorganisms and things. A eukaryotic cell typically is between 10 micrometers and 500 micrometers. Now, obviously, the largest cell you've ever seen, a chicken yolk cell, a chicken yolk, is much larger than 500 micrometers. And a prokaryotic cell, it can be smaller than 1 micrometers, and the largest is about 10 micrometers in size, meaning the largest prokaryotic cell is actually larger than some eukaryotic, but up to the size of your average eukaryotic, not your average, your small average eukaryotic cell, meaning the small range here. And then a virus is about 1/10 the size of a prokaryotic cell. The very largest virus is actually up to the size of the smallest prokaryotic cell, and that's 440 nanometers or 0.44 micrometers. The smallest prokaryotic cell that they think is viable is zero point 0.4 micrometers in diameter. And you don't need to know that. I'm just telling you the smallest cell that they think is viable. There are smaller prokaryotic cells, but people think they're not viable. And I'm not sure how you would measure that, so we're not going to get into it. And then the smallest virus goes down to about 0.02 micrometers. That would be 20 nanometers. The very largest virus we're going to talk about is megavirus. Ebola virus in its length is actually 1 micrometer in length, but its diameter is less than 0.44 micrometers because it's very long and skinny. So megavirus is the largest virus by its diameter. And then the smallest virus we'll talk about is actually drawing a blank now on what it's called. Jeez. Ah, it's coronavirus. What the heck is that? Causes a very serious disease in childhood. Fortunately, it's only in 3 countries in the world at the moment, and it is, I'm losing my memory. It'll come to me. Just let me get back to that. You should know this graph here showing you the different sizes of things. The largest thing we're really going to talk about is a person, and you can't get things larger than a person, as you know. And then there's a chicken egg. That would be the yolk, the largest cell you've ever seen. The largest cell on the planet currently is the yolk of the ostrich egg, but I doubt any of you have ever seen that. I have had one student who said that she saw a turkey yolk cell. That would be larger too than a chicken yolk, but I've never seen a turkey yolk cell, and I imagine most of you haven't either. Frog eggs are about the smallest thing the human eye can see without the use of a microscope. And then things smaller than a frog egg, you need the light microscope to see a eukaryotic cell, a prokaryotic cell. Things smaller than a prokaryotic cell, you need the electron microscope to see. And that would include viruses, large proteins, large lipids, and then small molecules would be something smaller than that, such as the water molecule. And then atoms are the smallest thing we're going to talk about. There are parts of atoms, but we're not going to talk about them. Well, actually, I guess I do talk about electrons, so that's smaller than an atom. And you need an electron microscope to go down to the size of an atom. Any questions about any of this? Note that This graph does have an error here. You don't need to know the size of things. You only need to know the relative rank. The error in the graph is it's got 1 micrometer there and 1 micrometer there. I put in a correction there. That should be 1 millimeter right here. Any question about any of this? Still trying to remember that virus. Polio, there it is. I finally got it. The smallest virus we'll talk about is the polio virus, and I know you've all heard about the polio virus. All right, so this slide is showing you a light microscope, and it is a compound light microscope because it has two lenses to magnify the specimen. It has the objective lens here, which magnifies the specimen, and an ocular lens here that magnifies the specimen. The total magnification is the magnification of the objective lens times the magnification of the ocular lens. Typical microscopes in the lab have the objective lens of a 4x lens, a 10x lens, a 40x lens, and a 100x objective lens. And they do have different names, like the scanning lens, the oil immersion lens. I don't tend to use those names. You just take this magnification, and then for all of the microscopes in the lab, we only have 10x ocular lens. So you multiply this objective lens times 10x for the ocular lens. Now, the most expensive and the best microscopes will have a 20x magnification lens in the ocular lens. We do not have the best microscopes or the most expensive microscopes at Clark College. Any question about any of that? All right, so you should know the body parts of the microscope um, labeled here. The ocular lens, also called the eyepiece. You don't really need to know the tube, that's just holding the microscope together. The arm is where you hold the microscope. The objective lens right here, this one has four. The stage, which has the slide on top of it. And then there's stage controls. You should know that too. And that's for moving the slide around and holding it in place. There's the condenser lens right there and the iris diaphragm right there. I think in the previous picture I showed you in the lab, the iris diaphragm was above the condenser lens. And I don't know why in some microscopes the iris diaphragm is above the condenser lens and others is below the condenser lens, but the two are always together. The condenser lens just gives a cone of light up to where the specimen is, and then the iris diaphragm is like a shutter and can open up that cone of light or close it down. The illuminator, also called the lamp or the light source here. The coarse focusing knob, the big one, the fine focusing knob, the small one, and then the base is where you put your other hand for supporting the microscope. Any question about any of these parts? Now, this is a simple microscope. It has only one lens for magnifying. So a simple microscope is really just a super duper magnifying glass. Magnifies only one time with one lens. And this is the microscope that what the heck is his name? Um, Not doing too well today, remembering things. uh, Van Leeuwenhoek made. A simple microscope because it only had one lens, and of course, this is also a long time ago that he made it, and so it's a very simple microscope. Most simple microscopes built today only have one magnification lens, and they would be similar in design to this, but they would only have an objective lens, and they don't have a lens up here. They would just have one tube usually that you have to look through, and no lens there, magnifying lens. Anything else I need to talk about that? No. Resolution is the ability of the lens to distinguish two points as separate points. The microscope has a resolving power of 0.02 micrometers, and this is the smallest two points can be separated from each other and be seen as two distinct points. And that's because a light microscope cannot magnify any more powerful than this, meaning you can't resolve anything smaller than 0.02 micrometers apart because of the properties of white light. The resolution of white light is around 2.0, no, zero point 0.2 micrometers or 200 nanometers. And that has to do with the wavelength of light. The best compound microscope, like I said, can magnify 200,000 times. And if they were to make a scope that would be higher magnification, it would be empty magnification because 200,000 times gets down to the resolution of zero point 0.2 micrometers or 200 nanometers. And you can't magnify any further. It would just be empty magnification because it would not see 2 points as separate 2 points smaller than 200 nanometers apart because of the properties of white light. Any question about any of that? Now remember, our microscopes, the highest magnification they can give is 1,000 times total magnification. And you can get better than that. This would be two separate points, and there you can see them under the light microscope. You can make them out as two separate points. These right here are less than 200 nanometers from each other, and you cannot make them out as separate points. And that's what resolution is. Any question about any of that? All right. Light bends whenever it comes out of one medium and enters another medium. Like when light comes out of the glass slide and then enters the air, the light will bend at the interface. And that's why the pencil looks bent right there because of the bending properties of light. And it happens right there at the interface. And they're trying to show you the artist rendition there. That is actually a straight pencil, by the way. And it's looking this way just because of the refractive properties of light. Light bends when it comes out of one medium and enters another. This refraction of light is actually very important for life. For example, a fish in the water will look like it's in this position to the bird's eye. And the bird instinctively knows that the fish is not at this position. It'll put its feet down lower to catch the fish, which is actually in this position. And what happens is the light coming through the water will then bend as it enters the air. And that's why the fish will look like it's in this position, but it's really in this position. And like I said, birds are used to this and they know that the fish is not in that position. They'll put their feet down lower. One advantage of using oil is that light coming through glass and then entering the oil will not bend because oil has the same refractive index as glass. And therefore, if you use oil, the light coming out of the glass slide will not bend. Now, if you're sending light through air, the light coming out of the glass slide will, or at least some of it, will bend. This light ray, as you notice, bends so hard it's sent back into the glass. That one is bent hard enough that it will not be captured by the objective lens because this light ray is bent away from the objective lens. That one is bent away, but it is not bent away far enough that it cannot be captured by the lens. And this one happens to not be bent at all, and so it will be captured by the objective lens. Now, if you're using oil, all three of these light rays will be captured by the objective lens, and in the air, only two of them, this one and that one, will be captured by the objective lens. So if you're using oil, more light rays will enter the objective lens, and then your resolution of the microscope will be better. So you get greater resolution if you use oil, and it's simply due to the fact that you're capturing more of the light rays. Any question about any of that? And that's why we use oil with the 100x objective lens, so that we capture more light and then get better resolution than if we did not use oil. So oil has the same refractive index as glass. Oh, and I should mention that this light ray here actually bends two times, coming out of the glass here, and then entering the air. And then when leaving the air and entering the glass of the microscope, that one is actually refracting twice. And these aren't refracting at all. So if you use oil, you'll get better resolution because the light will not refract when coming out of glass or entering glass because oil has the same refractive index as glass. Any question about any of that? Remember, the refraction of light is just the bending of light. All right, this slide is showing you the difference between bright field illumination and dark field illumination. Bright field illumination is what the microscopes in our lab use, and most microscopes use bright field illumination, where you have a light source down here, and the light goes through the condenser lens, put onto the specimen, captured by the objective lens, and then you see the specimen with your eye. The specimen is lighted up, and then the background of the slide is very light because all it is white light coming through the glass. Any question about bright field illumination? Dark field illumination differs from bright field illumination in that we have a disk in between the light source and the specimen. And that's why the background of the slide is dark because there's no light coming from the light source. It's blocked by the disk. Now, some light at an angle comes through the condenser lens and then comes to the specimen, but then the specimen refracts the light and can refract some of those light rays into the um, objective lens. And so the specimen is lit up, but it is darker than the specimen under bright field illumination. The glass slide, on the other hand, does not have the refraction of light, bending the light into the objective lens, so the glass slide looks dark. And this is why we call it dark field illumination. You get no light directly from the lamp. The only thing you're seeing, the specimen, is some light rays which are bent by the specimen themselves into the angle, the correct angle to be captured by the objective lens. You do need to understand bright field illumination and dark field illumination. You only need to know the concept. Okay? Any question about any of that? Your book talks a little bit more about bright field illumination and dark field illumination if you care for a deeper explanation. Here we're looking at the electron microscope, and you can see it's much bigger than a light microscope. An electron microscope uses a beam of electrons instead of light to see the specimen. The beams of electrons are not focused by a glass lens. They are focused by an electromagnetic lens. And what they're doing is they're using the electromagnetic lens to bend the electrons so that you can see the specimen. Electrons have a shorter wavelength than light, and so you can see things 100,000 times smaller than you can see with visible light. And that's because electrons can give a greater resolution. So an electron microscope gives greater resolution than a light microscope. Any questions? All right, not hearing anything. So let's move on. Here is two specimens using an electron microscope, showing the two main types of an electron microscope. Transmission electron microscope, shown here, and a scanning electron microscope, shown here. Let me blow those up. And what can you tell me about the differences between the two types of a microscope? Come on, you gotta participate. What's different between this one and that one? There's two things. Obviously, one you should see very easily. No one wants to contribute. Well, we're just going to move on then. Oops. Oh, shoot. What the heck happened there? A little larger. Out there. All right, you will be quizzed on that. Here you're seeing two specimens again. This being a scanning electron microscope, and that being a transmission electron microscope. Anyone see a difference? All right. The third type of electron microscope you're not going to be quizzed on. I'm just going to mention it. It has a resolution of 1/100th of an atom. It is called a scanning tunneling microscope. Uses a metal probe to scan a specimen. Go that down a little bit. And then this is the fourth type of a microscope. You will not be examined on it. It is the fourth type of electron microscope. It uses a metal and diamond probe inserted into the specimen. It produces a 3D image of an object. Here we're looking at the toxin from Clostridium perfringens. And I guess I should mention the scanning tunneling microscope is the most we can magnify any specimen, meaning usually we see something smaller than an atom. It is using a scanning tunneling microscope. You won't be examined on that. I'm just mentioning it. Any question about the electron microscope? All right. This slide is showing you cells. Which slide is easier to view? The one on the left or the one on the right?
The one on the right?
Yes. The only difference between these cells and these cells is these cells have been stained. That's the only difference. And that makes the cells much easier to view. Staining, all it does is increase the contrast between the specimen, shown here, and the background, shown there. And that's all staining does, increasing the contrast between the cell and the background of the slide. Cells are mostly water, and water, as you know, is very transparent to light, and that's why these cells don't show up much differently than the background. So cells have little contrast with the background of the slide. They're difficult to observe. And if you want to observe them better, you use a stain. And all you're doing is increasing the contrast between the specimen and the background of the slide, which for most cases is the glass of the slide. Any question about any of that? With bright field microscopes, which is most microscopes in most labs, microbes are colorless and have little contrast with the slide or the medium. And that's most microbes are colorless. Obviously, if you're looking at algae, it will be most of them green, some of them red, some of them brown. But the point is, most microbes are colorless and you can't see them very easily against the background of the slide. To see them well, you need to use a stain. Staining is coloring the specimen or coloring the background of the slide to increase the specimen's visibility simply by increasing the contrast between the specimen and the background of the slide. A smear, like a bacterial smear, is just a thin film of cells spread on a slide. When you make a smear, you are not staining the slide, although most bacterial smears will later be stained. But the important part to know is that making the bacterial smear and making the stain or staining the smear are two separate events. So remember that making a bacterial smear is not staining the cells, does not involve staining the cells. It is a separate event. Students always get that confused. But making a bacterial smear is a separate event from staining the cells. So making a bacterial smear is just putting the cells, usually in a thin layer of cells, on a slide and usually you want no more than one cell layer thick. So prior to staining the microbes, you make a bacterial smear. After you make the bacterial smear, it's actually part of making this bacterial smear. You then fix the cells to the slide to attach them. It kills many of them. It doesn't kill endospores, doesn't kill all of them, but it kills most cells other than endospores. And you usually attach them with heat, and that makes the slide, the cell, attached to the glass slide, and then it doesn't wash off in a later step, which is usually smearing the cells. I'm not smearing the cells, staining the cells. Usually you make the bacterial smear, and then you later stain the cells, and the staining process itself can wash off the cells if they're not fixed onto the slide. Any question about any of that?
For the smear, would it look like the left slide in slide 21, and then the stain would be the one on the right? Like, is that kind of an
Uh,
example?
Not exactly. What we're looking at here is this is a wet mount, and the cells are not stained. They're just in water on the slide. Okay?
Hmm.
And this probably is also a wet mount, but the cells are stained. So the cells are in the same water, but they are stained so you're seeing them better.
Oh, I
Okay.
see. Uh-huh.
Usually, when you're looking at a smear of bacteria, you will not have it a wet mount, and the cells will be stained and then dried.
Oh, I
Okay.
see. How do they dry it without taking off the cells like you mentioned earlier?
You simply let the water air dry, meaning evaporate within a few minutes, usually
Oh, okay. And
air
this would
dry, and
be,
then you heat fix it to fix them to the slide.
oh, I see. And then would you still stain if it was a dark light microscopy?
Meaning dark
Is that,
field microscopy.
yeah, dark field.
Oh, I've never used dark field microscopy, but yes, you still do stain. In fact, if you take a look at the cells there, they are stained because they're staining purplish blue. And cells won't have that color. They'll be clear if they're not stained. This cell is stained as well.
Oh, I see. Okay, thank you.
Sure. Good questions. All right, I think that's it there. So with bright field microscope, which are most microscopes, the microbes are colorless, and to see them well, you need to stain the cells. Staining is just coloring the specimen or the background to increase its visibility and the contrast between the cells and the background of the slide. A bacterial smear, I've already mentioned this, is just a cell, thin layer of cells put on the slide, usually one layer thick. And you make the bacterial smear prior to staining the cells. You do not need to stain the bacterial smear, but oftentimes we do. You let the bacterial smear then air dry, usually just a few minutes, and then heat fix them to the glass. And I'll go over that when I'm talking about the gram stain, how you make a bacterial smear exactly. But you just mix the cells on some water on a slide, spread the water all over the slide, let it air dry. And then once it's air dried, you slightly heat the slide, where you just warm the slide. You do not make it hot. And that's to heat fix the bacteria to the slide, attaching the cells to the glass. Stains, also called dyes, are salts consisting of a positive and a negative ion. The dye will be in one part, either the positive or the negative ion. And we call the dye the colored portion, which we also call the chromophore. And a basic dye has the chromophore on the positive ion of the salt, or the cation, the positively charged ion. Acidic dyes has the chromophore in the anion, the negative part of the salt. Any question about that? So this tells you we have two different types of stains, a basic dye and an acidic dye. The basic dye has the colored portion, the chromophore of the stain, which colors the specimen, on the positive part of the salt. Most stains in microbiology are basic dyes, but we do have some acidic dyes. Bacteria have a slightly negative charge. Therefore, a basic dye is generally better for staining the cell because the negative chromophore will be attracted to the positive chromophore of the basic dye will be attracted to the slightly negative cell. And then the cell will stain the color. Some basic dyes include crystal violet, methylene blue, malachite green, and safranin. And we'll talk about all of these different dyes. Any question about any of them? A acidic dye has the chromophore in the negative part of the salt. The negative part of the salt will be repelled from the negative cell. And so the acidic dye will not stain the cell, it will stain the background or the slide of the cell. And some acidic dyes include eosin, acid fuchsin, and nigrosin. And we will hardly talk about any of these. We might talk about acid fuchsin one time. But the important part is an acidic dye stains the background of the cell, not the cell itself. And this is called negative staining because in the old Polaroid film, you had a negative and things were colored, or actually not colored, but um, illuminated the reverse of what the specimen was, meaning the negative where the object was very light would be very dark in the negative, and then where the object was very dark in the negative, it would be very light. Any question about any of that? Here are the two examples of a basic stain, staining the cells, and then a negative stain, staining the background of the slide, not staining the cells. Both of them illustrate the cell very nicely. Any question about any of that? So this is a basic stain. This is an acidic stain, and usually we call that a negative stain because the background stains, the cell itself is light, like a negative to a Polaroid film, if you know what that is. If you don't know what that is, don't worry about it. Any question about any of that? Both of them show the cell very nicely. All right. In microbiology, there are three main types of stains: a simple stain, a differential stain, and a special stain. A simple stain is a solution of a single dye. So one stain is in the preparation, like malachite green, and then it stains all cells that same color. The primary purpose of a simple stain is to simply increase the contrast between the background of the slide and the specimen. With a simple stain, you can determine shape of the cell, and that's about it. You can't see any differences in the cells because all cells will be stained the same color by the one single dye. Normally, when we are staining the cells, the stain will be applied to a bacterial smear. So you have to make the bacterial smear before you stain the cells. You then put the stain on for a certain length of time. It may be washed off. The cells may be dried and then usually heat fixed and then examined. When you're using a simple stain, you don't have to go through all of these steps because a simple stain, well, it's simple. You could just apply the simple stain directly to a wet mount, in which case you're applying the bacteria to the wet mount. You're not washing the cells off. You're not drying the slide. You're then adding the stain and then examining them. Okay, so that's how it could be different. And like I said, with a simple stain, it's very flexible how you're using it. You may use a mordant. It will intensify the stain. The functions of a mordant are, first of all, it's not a stain itself. It simply increases the affinity between the stain and the specimen. Like the mordant in the gram stain simply makes the crystal violet stand out better than if you only had crystal violet alone. The mordant is iodine. We'll talk about that in the gram stain. A mordant can also simply coat the specimen, enhancing it, thereby making it much easier to see. For example, let me blow this up. Here we're staining the cells and then applying a mordant, coating the cell, and actually you're applying the mordant several times. And what it does is it puts many layers down so that you can see the flagella on the cell. If you didn't put the mordant and actually many coats of the mordant on, you would not see the flagella because the mordant um, is putting on a layer each time you apply it. Any questions about that? We'll talk more about the mordant when we're talking about the gram stain in a little bit. A differential stain is a stain that reacts differently with different kinds of microbes, thereby allowing you to distinguish between two different types of microbes. Usually, the two cells will take on two different colors, and usually you're using two or more stains in a differential stain. It is not required that you use two or more stains in a differential stain, but most differential stains do use two or more stains. Any question about any of that? All right, the two most commonly used differential stains in microbiology are the gram stain and the acid-fast stain. We'll talk about the gram stain in depth in this lesson. And the acid-fast stain we'll talk about a little bit later. All right, the gram stain was developed in 1884 by Hans Christian Graham, Dr. Graham, and is named after somebody, so you should capitalize the G whenever you write the gram stain or gram, like gram negative. You should have that G capitalized because it's named after somebody. You don't need to know when he did it, but you should know that Dr. Graham invented this stain. It was then used by microbiologists, and they found it a very useful stain because it classified bacteria into two very large groups, the gram-positive cells and the gram-negative cells. Now, Dr. Graham was using it as a differential stain, and he was using it to stain gram-positive cells, which stain purple, to use it to highlight the bacteria were different from the patient's own cells, which stain pink. So Dr. Graham was using it as a differential stain to differentiate the patient's cells from the bacterial cells. But microbiologists noticed that it was a differential stain, separating all bacteria into two large groups, the gram-positive cells, which stain purple, and the gram-negative cells, which stain pinkish. The gram stain, in brief, is you get a bacterial smear, you let it air dry, heat fix it, and then you take that bacterial smear and you use it for your gram stain. You first flood the slide with crystal violet, wash the crystal violet off with cold water, flood the side with iodine. Iodine is not a stain, it is a mordant. Wash it off with distilled water, then flood the slide with acetone ethanol. This is the decolorizing agent. You only do it briefly for about 5 seconds to remove the crystal violet iodine from the gram-negative cells, but not from the gram-positive cells. You then wash immediately with cold water, then flood the slide with safranin. It's the counter stain. It will stain the gram-negative cells pinkish. You then wash it off with cold water, let it dry under with bibulous paper, and then look at it under the microscope to look at the cells. Any question about the basic procedure? Now, when you're giving me the basic procedure, you do not need to state that after each step, you wash with cold water. You just need to state at one place, either in the start or at the end, after each step, you wash with cold water. Any question about that? When we're applying the gram stain, when you apply the crystal violet, all the cells will pick up the crystal violet and will look a bluish purple color. You then apply the Graham's iodine, which is not a stain. It complexes with the crystal violet, making a larger complex than the crystal violet or the iodine alone, so that it will intensify the stain. That's not shown in this slide, but the cells will appear a little brighter, a little darker, and the color is a little different. Instead of a blue purple color, there'll be a purple purple color, and that is not shown in this slide. You then apply the decolorizing agent, and it will remove the crystal violet iodine complex from the gram-negative cells, and they will appear clear, but it does not remove the crystal violet iodine complex from the gram-positive cells, so they will appear purple. So the decolorizing step is the differential step of the gram stain, because this is where the cells have two different appearances, two different colors, so to speak. Although that's not really a color, but it's two different. We're just going to leave it that way. And then you apply the safranin to add color to the gram-negative cells to make them stand out from the background of the slide. And then after the safranin, the gram-negative cells will be pinkish in color. The gram-positive cells will be a purple color. Any question about that?
With the differential stain and like the gram staining procedure, kind of what would be used if they were like studying a virus or a disease?
Well, a virus you can't see with an with a light microscope. Um, you do stain cells with an electron microscope, but it's a little different than the gram stain. Um, so I'm not sure how to answer your question. You do stain cells with an electron microscope. You do with a light microscope. Does that answer your question?
Yeah, it does. I was just curious if they had any like correlation to that.
Okay. Hopefully I answered that, and I'm not sure. uh, let's move on. Um, so, to do the gram staining procedure, you must have a bacterial smear. The bacterial smear should be air dried and then heat fixed so the cells are attached to the slide. If you do not air dry and heat fix the cells, which will attach the cells to the cells will wash off simply by putting the crystal violet iodine on. That will lift up the cells. And then when you dump the crystal violet iodine off, you wash off most of the cells. And then in your next step, where you're washing the slide, where do I have that? Oh, there it is there. When you're washing the slide with water, you'll definitely wash off all the cells. So you must have the bacterial smear air dried and heat fixed before you can do the gram stain. Any question about any of that? The bacterial smear is a preparation you do before doing the gram stain or any stain. You then take that bacterial smear and then do the gram stain. First, you flood with crystal violet. You flood with crystal violet for 1 minute. After that, you dump off the crystal violet and then gently wash the cells in cold water. Remember, after each step of the gram stain, we'll have a cold water wash. Crystal violet will stick to the peptidoglycan in the bacterial cell, and the bacterial cell wall has the peptidoglycan. It's considered the primary stain because it colors all cells the same, and it'll be a bluish purple color. So here, both the gram-positive cells and the gram-negative cells are bluish purple in color. You then, after you've washed off the crystal violet, and you dump off as much of the water as you can. You don't need to get all of it off. You then flood the slide with iodine for 1 minute. Iodine is not used as a stain here. It is simply a mordant. The iodine will complex with the crystal violet, creating a larger complex in the bacteria cell that will make the cell slightly more visible, and then the color will be slightly more purple rather than bluish purple. You then dump off the iodine and then wash with cold water. Like I said, iodine here doesn't stain the cells, it's a mordant. It will change the color just slightly to a purple color. And then the cells will be a little more intensely stained. Iodine in the gram stain is not used as a as a dye, but if you remember from Biology 160, we sometimes stain plant cells with iodine because iodine will color the regions of a plant cell which have glucose and sucrose in them. So iodine could be used as a stain, but in the gram stain, we're not using it as a stain. We're using it as a mordant. All right. You then dump off as much of the water as you can. You don't need to get all of it off. And you add the um, decolorizing agent, which is usually acetone ethanol, but there are different decolorizing agents. It could be acid, ethanol. It could be also just ethanol itself. And what you're doing when you're applying the colorizing agent, can you guys see this here? Let me stop the share and then I can see it myself. There, you can see it there. So this is the slide with the cells. What you do is you add the decolorizing agent on the top of the slide, watch it run down. Once the crystal violet stops running, stop adding the decolorizing agent. And that will happen in about 5 seconds. And that's the best way to add the decolorizing agent. Add it while you're watching, stop adding it once it stops running. Any question about any of that? Come on, share. So you then wash the slide immediately with cold water. Why it needs to be an immediate wash here is the trace amounts of decolorizing agent on the cells will continue to work. And if given enough time, the decolorizing agent will remove the crystal violet iodine complex from the gram-positive cells. And that's why we apply it for only about 5 seconds, and then you wash immediately to remove the decolorizing agent from the cells. If you do not do that, you will get the decolorizing agent removing the crystal violet gram iodine complex from the gram-positive cells, and you don't want that to happen. Any question about any of that? So all the other steps, you don't need to wash the cells immediately, but on this one you do. After applying the decolorizing agent, if it's done correctly, the gram-positive cells will stain a purple color. The gram-negative cells will be colorless or unstained. And that's assuming you're applying the gram stain correctly. If you, for example, did not apply the decolorizing agent for long enough, then the gram-negative cells will be stained purple. And if you apply the decolorizing agent for too long, the gram-positive cells will be colorless as well as the gram-negative cells. So that's why the decolorizing step is very tricky. It's the hard one. And practice usually allows the student to get the gram stain, meaning how to do it. All right. Then you immediately wash the slide with cold water, dump off the water, and then you apply safranin. Safranin is a pink dye. It does stain all of the bacteria. We call it the counter stain because it has a different color to the primary stain. So crystal violet is the primary stain, safranin is the counter stain, and in my example, you apply all the ingredients except for the decolorizing agent. You apply all of them for 1 minute. Usually the safranin is strong enough that 1 minute is good enough. I used to work at PCC, Portland Community College, where the people who made the safranin, occasionally they would make the safranin too weak, and 1 minute would not be good enough time to apply the safranin. You had to do it for 2 minutes. But the point is that if it's made correctly, which at Clark College it always has been, 1 minute will be good enough. You then dump the safranin off and then wash the cells in cold water. The gram-positive cells, if the gram stain was done correctly, will stain purple. The gram-negative cells will stain pink. You then take the slide and you simply blot it dry with bibulous paper. Let me talk a little bit about bibulous paper now. It's a very thick paper, sort of similar to thin cardboard that you can easily bend. You rip it out of the book, take your slide, fold over the bibulous paper. I don't know if you can see this or not. So you fold it over, and then blotting it dry just means press down on the bibulous paper to blot the slide. You do not take the bibulous paper and slide it against the slide. You do not take the slide and slide it against the bibulous paper, because if you do, you would remove the cells. You just blot it dry, where you press down the paper directly onto the slide. And then enough of the water is removed that you can examine the slide directly under the microscope, even if you're using oil and 100x compound, no, the objective lens, 100x objective lens. Any question about any of that? All right, the gram-positive cells are purple, the gram-negative cells are pink after the procedure. Why? What is the basis of the differential staining? Well, first, let me get back to this point here. And then I'm stating that the safranin stains all of the bacteria. Why do the gram-positive cells not stain pink? Since the safranin is getting in the cells. Anyone have a guess? I really hate that none of you like to guess, but I will tell you this one because you shouldn't necessarily know this one, and that is the safranin gets in the gram-negative cells because that's the only stain in these cells. The crystal violet iodine complex was washed out in the decolorizing step. However, the cells, the gram-positive cells, have the crystal violet iodine complex in them. They also have the safranin in them. But to our human eye, we only see the purple stain because purple is a stronger color. The human eye only sees the purple color. If you were to look at the cells, so let's make this a cell under an electron microscope, you would see one part of the cell purple and another part of the cell pink. So the safranin is getting in the cells. It's just that our human eye cannot see the safranin because it is hidden behind the purple color. Any question about any of that? So the safranin is in the gram-positive cells. We just cannot see it because of the way our eyes see color, I guess. And it's just that pink doesn't show up to us. But if you were to look at it under the electron microscope, you could see pink in the cell as well as purple in the cell. But why do the gram-negative cells stain pink and the gram-positive cells stain purple? What is the basis of the differential staining? It's because the gram-positive cells have a thick peptidoglycan layer. This thick layer of peptidoglycan retains the crystal violet iodine complex. The gram-negative cells, if they have a cell wall, have a thin peptidoglycan layer. This thin peptidoglycan layer is not thick enough to retain the a crystal violet iodine complex. So it washes out of the cell. The lipopolysaccharide layer is also destroyed, meaning the cell wall is also destroyed by the acid alcohol wash. It will remove the destroy the lipids of the cell membrane. That's not terribly important, but it will happen, which allows the crystal violet, not the crystal violet, safranin, to more easily enter the cells. So the gram-positive cell shown here, let me blow this up, has a very thick layer of purple, a very thin layer of yellow. The thin layer of yellow is actually the bilayer of the plasma membrane, which is mostly phospholipid molecule, and has a thick layer of peptidoglycan. Shown here, it's only showing 3 layers of peptidoglycan, but in reality, it's something between 20 layers of peptidoglycan and maybe 100 layers of peptidoglycan. It's very thick, the gram-positive cell, and how many layers of peptidoglycan it has. The gram-negative cell, on the other hand, has a thin yellow layer here, a thin purple layer there, and then a thin yellow layer there. That thin yellow layer on the lower side, once again, is the cell membrane, the lipid bilayer of the cell membrane. The thin layer is peptidoglycan, which is shown as only one layer. Don't be mistaken by that. The peptidoglycan of the gram-negative cells is a couple of layers, between 3 and 20 layers of peptidoglycan. So it's more than just one layer. It's at least 3 and up to about 20. But it's thin, which is why the crystal violet iodine complexes washes out of it. And then on the outside of the gram-negative cell, we have the outer membrane of the cell wall, and it is a lipid bilayer, similar to the cell membrane. Phospholipid is actually all of that layer of the cell membrane, and it's a large portion of this layer, but there are other lipids in this layer of the outer membrane. Any question about any of that?
No, but it's 6:20.
Oh, thank you. Let's go ahead and end it then. How many slides do I have here? Just a couple more. Once again, showing you the thick layer of the gram-positive cell, the thin layer of the gram-negative cells. So we'll stop right there. All right. Any questions before I go? All right, I'll see you in the lab at 6:30.